This may be changes in the anthropogenic loading of nutrients into the water column, preferentially in the source waters that propagate into our water masses. That's a hypothesis. It's something we can test. And that productivity drives carbon flux. This is the sediment trap measurements of carbon flux at 150 meters. Also has that same seasonal cycle. It looks just like primary productivity as well. In this case, there's really not much of a trend. So if productivity is increasing but flux isn't, we're either missing something or somewhere in the water column, biomass is building up over time at unrecognizable rates. We've also looked at thermohaline trends. This is looking at the depth dependent in the top 1,200 meters, changes in potential density, changes in salinity, changes in potential temperature as well. And you can see here in blue that we've got a sort of peak warming actually at 150 meters. So you've got exchange of heat with atmosphere that sort of caps this water mass here that has been warming over time, about 1/4 of a degree per decade. We've got a little bit of freshening in the thermocline in this region, and these really are driven by changes to evaporation precipitation balance, again, which is driving our density changes. So the waters are changing. Now, this is one that we understand very well. What is driving the increase of CO2 in the ocean? That's what you see here in blue. This is what I call the Aloha curve, where we're seeing the long-term increase in the partial pressure of CO2, and that matches the change that we see at the Mauna Loa Observatory. It's on the Big Island of Hawaii, where they've measured increases in CO2 in the upper atmosphere. CO2 increases in the ocean, in the atmosphere, CO2 increases in the ocean, and that drives a very predictable change in pH. This is the concept called ocean acidification. This was first reported by John Doerr in 2009, and obviously we keep these time series up to date. This is measurable, real, persistent change. And that change is not confined to the surface. Here we see the changes in pH and DIC, dissolved inorganic carbon, with depth. These alterations of carbon chemistry have propagated to the upper 600 meters of the water column, and there's apparent acceleration of acidification in the mesopelagic at these sampling horizons of 350, 500, and 600 meters, with the maximum rate of change at 500 meters. And this is nearing 0.004 units per year, which sounds, small, but it's equivalent to a 33% increase in hydrogen ion concentrations over the time series at the mesopelagic depth horizons. This accumulation rate is 5 times what we've observed in the surface, and again, may be reflective of conditions where these water masses are formed in the far Northwest Pacific. So here's a chance for us to understand, again, I just want to come back to primary productivity because it's one that we understand quite a bit less. So this 30-year increase in productivity, it's about 37% increase. There's a 17% increase in chlorophyll A, as well as an 8% increase in biomass. Again, we hypothesize that these are resulting from enhanced supply of nutrients to the lower portion of the water column. It's a working hypothesis. There may be other explanations. Perhaps we might even see these rates of productivity being linked to those changes in CO2 that I just showed. But I showed that again just to get this point here. These long-term trends are not captured by satellite-based models. So in black with the white circles are the data from the hot time series. Here in orange is data from the VGPM satellite. It's a chlorophyll-based model. Um, this is the cafe model, which is both the cafe and the carbon-based model are really looking at the ratio of carbon to chlorophyll and interpreting that as a driver of the rate of net primary productivity, which is what we're measuring with 14C. And you can see hot shows an increase and every single model shows a decline. So mechanistically, these models are either not capturing, in most cases, don't capture the variability nor the trends in the rate of productivity, or there could be perhaps some issues in the recovery of those products that are used to drive the productivity models. There's been a blend of satellites over the years. So what drives productivity and export in gyres? This gets back to the lovely intro you guys gave to me. Nitrogen fixation is a player. It's been estimated to support about 50% of particle export at Station Aloha. We have been measuring this rate since 2005. There's very weak seasonality. Highs are generally measured in our summer to fall months where it's warmest. And we don't understand the drivers of this metabolism, nor what controls the functional diversity of the causative organisms. We do know that nitrogen fixation is at least partially responsible. What you're seeing here is what we call the summer export pulse, where we get a nice increase in the magnitude of carbon flux, nitrogen flux, and silica flux to the deep sea. These are bottomward traps in summer that seem to be driven by these diatomic diazotroph assemblages. I'm sure you've heard this from Rachel, but there's many, many things we do not know about the diversity, activity, and controls of nitrogen fixing organisms. What we do know is they seem to drive consistent blooms. These are basically they're from 2004 to 2019 in the summer months. And what you're seeing in the green is an outline for a proxy that we're using to look at deviations of chlorophyll from satellite maps. Chlorophyll is not perfect; doesn't always tell you, really what the biomass is there because there's a strong photoacclimation signal. But when we start to look at seasonal spatial deviations, we can start to map that out. It's a little bit hard to see, but here in black is the islands of Hawaii. And again, these blooms are very classically centered around 30 North. But you can see years like 2018 when we had the eruption of Kilauea, these absolute massive blooms that are in orders of 1 million square kilometers in area and lasted for 2 months at a time and are thought to be driven by. These are examples of nitrogen fixing organisms that perhaps are driving these surface expressions of increased phytoplankton biomass. They're not small. These blooms are not small. They're not isolated. They're considerably large in their spatial and temporal footprints. So what do we think is driving some of these climate oscillations? This is work that was published not too very long ago by Ricardo Letelier and others, including myself, where we looked at the Pacific Decadal Oscillation, and this is satellite-based dust aerosol optical depth. And what you can see here is there's a strong correlation between the PDO and the input of dust, specifically to Station Aloha, likely that is driving some iron deposition. So we think this is a strong potential driver of these aperiodic summer blooms. The peak seasonal input of dust comes in sort of spring. This is derived from mostly anthropogenic burning from Asia. The last few things I want to say about nitrogen fixation, again, it's something that we very much want to understand here, what controls the process, what controls its variability. Now we're able to start measuring the mesoscale abundances of these organisms. This is the depth distribution of nitrogen fixation with the positive red plus symbols as our outliers. And we see some of those outliers really being driven by what we're calling mesoscale eddies. This is a satellite chlorophyll map of of a mesoscale eddy with the sea surface height anomalies. So we're seeing strong abundances of organisms. We've been using imaging flow cytometry, which is what you have here in the last, I think, 4 years now. We've had an imaging flow cytometer on all of the hot cruises, and that allows us to really get a better handle on the sorts of organisms that are blooming and thriving here. And we are just now starting to look more finely into flow sorting of these organisms. This is recent support from NSF to Julie Granger at the University of Connecticut and Adrian Marchetti at University of North Carolina, Chapel Hill. That's going to allow us to use isotope tracking, flow sorting, and metagenomics to more carefully evaluate the various sources of nitrogen that fuel productivity and export at this station. So to put these investigations into context, this is a recent paper by Shao et al. That's updated some data that we've had for a global distribution. This is version 2 of a global oceanic nitrogen fixer database. There's 90 co-authors on this. I'm pretty sure Rachel's on this too. And what you can see is, really what we know about the North Pacific is really driven by cruises into and out of Station Aloha. The North Pacific's about 46 teragrams of nitrogen per year. That's about a quarter of global nitrogen fixation. And the subtropical gyres account for 70% or more of all of the measured rates that we have across the global ocean. Okay, I'm going to move just quickly and shift directions in these last few slides to talk about some additional work that we've done at Station Aloha with support from the Simons Foundation. And just to give you some background, this is really the prevailing paradigm in oceanography of how changes in community structure drive changes in productivity. And this is called the Legendre's bifurcation model. The model is pretty straightforward. If you've got large phytoplankton, you've got fast sinking, and ultimately higher export efficiencies. Small phytoplankton, slower sinking, more microbial food loop. Hopefully you guys have heard about that, aggregation of cells, and higher ratios of recycled to total production, less total export. And this model is really prevalent in all of our satellite-based models of particle export. But in fact, when we start to look at the actual measurements, this was a cruise where we went from Oahu to about 35 north on 2 different cruises. And we looked at 2 different proxies, one based on forward scatter that gives you the particle size distribution, and another this imaging flow cytometer that allows us to image particles in the sort of 4 to 20 micron range and correlate that to, in this case, net community production in oxygen units, which is a proxy for export flux. And we found that, I'm not showing you the other size classes, but really it's this middle size class of nanoplankton that seems to drive net community production across these gradients in the North Pacific. So this bifurcation model is useful, but we're probably not capturing the importance of some of these middle size classes. So given this finding across ecosystems, we're trying to explore this question within a biome in our home region. And specifically, we want to ask the question of, can we measure complete size distributions of suspended and sinking particles and use this data to better predict particle flux? We know that there's clear partitioning of size classes within the water column that we can measure. This is an optical-based assessment for Station Aloha, where we looked at small cells, which tend to dominate at the deep chlorophyll maximum, which I'm happy to talk about if anyone is interested, and these sort of medium nanoplankton and microplankton that dominate within the surface mix layer. And so how are we going to do this? Basically, it's going to take an integrated approach, which is what we've begun at Station Aloha. We're combining traditional flow cytometry where you're looking at scattering and fluorescence of particles and then gating those particles to count cells, to these optical approaches that I just showed you a minute ago, to imaging flow cytometry. And now in the last 2 years, we've also been deploying a towed video plankton recorder on the cruises, as well as we have an underwater vision profiler, the UVP, which is on every CTD cast, that allows us to see everything from diatom mats to protists to mesopelagic fish. Here's just a few examples of what we can classify. This is using machine learning to take those particle abundances and to turn them into, to give them names, take pictures of things and give them names. So we can do trichodesmium colonies pretty well, and actually just on one of our last cruises, we saw millimeter length filaments of these organisms. We do marine snow, cnidaria, rhizaria, diatom mats, copepods as well. So there's quite a bit of information that we can get from this. We're also using imaging tools to interrogate what is sinking. Here's an example from the imaging flow cytobot where we've explored high resolution depth sequences of flux at Station Aloha and observed relative flux of non-fluorescent and fluorescent particles and traps spanning 175 to 500 meters. And we found that detrital material, if that's what we're going to call the non-fluorescent here in black, that doesn't attenuate as strongly. And importantly in this case, particles seem to have originated from surface waters, where we know we have suspended material that consists of large diatoms, DDAs, and other nitrogen fixing organisms, which you can see in these traps. We've got the frustules of diatoms as well as live trichodesmium in these deep trap materials. So through these combined tools, we really hope to understand and model particle flux, which so far all signs point to a revised paradigm of flux and the importance of a middle class of these intermediate sized organisms. This is an example of a larger class organism, but basically diazotrophs and medium-sized cells that seem to be driving some of our productivity. So I'm just going to end with a little bit of a shift. Our work is far, far from done. It's a work in progress. We hope that continued collaborations at Station Aloha with anyone that wants to come on these cruises will help us better understand these critical ecosystem functions. So we invite anyone to join us, students, postdocs. All you need to do is sign up to volunteer and make it out here to Hawaii. So what have we learned from Station Aloha? 35 years, we are just now unraveling seasonality, identifying drivers of anomalies, looking at the imprint of humanity on the ocean. We know that we have a warming, increasing CO2, lowering of pH that is related to anthropogenic change. But the controlling aspects of this ecology, what are the factors that drive and control productivity, nitrogen fixations, and contributions to export, are far less certain. We've had a number of what we'll call value-added programs, both our own work, but also work from all sorts of people that have used our open access data to identify questions and problems and secured funding to help us better understand these processes that are being observed at Station Aloha. So what about the rest of the ocean? I want to talk about the FAIR model. This is a data format model, findable, accessible, interoperable, reusable. That's the FAIR. So I want to talk about that I point, interoperable. We have recent funding from NSF to bring together members of the oceanographic, data science, and informatics community to start building consensus on key components of FAIR models. And that includes things that most people don't think about, like common vocabularies and metadata reporting standards and data citation practices. And our hope is that we'll include modelers, educators, and decision makers and start to broaden applications from marine time series data, as well as foster collaborations and regional networks. And so I want to talk about one thing that we've done with this so far. This is a consortium that was led by Nico Lang at Geomar. They generated a synthesis of essential ocean variables, dissolved oxygen, dissolved inorganic nutrients, inorganic carbon system parameters across a number of time series. And in preparation for this, we prepared metadata formats and naming conventions in a CF, a global CF format, that we hope will allow for future interoperability and discovery of our data, as well as time series around the world and the Pacific and the Atlantic as well, and one, in this case, that got included from the Southern Hemisphere. So I want to show one slide from that work, and I know this is busy, but basically here's a single parameter. This is alkalinity, and in this case, these are 9 different time series and their data, either in monthly anomalies or monthly means as well. And what you'll see is Station Aloha is up here in the corner. This is a very data-rich time series. And what we're able to do with this is, now that we understand the seasonality and the long-term variability, we can calculate the minimum trend detection time. So the minimum observational period needed to statistically distinguish between natural variability or noise and anthropogenic forcing. And in that case, in this particular case, this is 42 plus or minus 7 years, which is exactly the length of the time series. So we are literally just now at a place where we're able to really measure these long-term changes and the differences from the seasonality, separate the signal from the noise. So what can we address with coordination of time series, large and small? I would say everything from methods to global and regional change, as well as uncertainty. Now, over a century of observing Earth's biogeochemical cycles, we still lack, I'd say, fundamental knowledge about the controls of these essential functions. And that knowledge in time series like HOT are really essential for understanding the response of the Earth system to human perturbations. There are parts of global change that we know with a very high level of certainty, ocean acidification, for example, and those we have very poor mechanistic understanding and a high uncertainty, export, for example. And here are 2 images from work that was done by Paul Quay, Steve Emerson, and Hilary Pavlevsky that found that the regional variability in observed export is pretty modest, about 3-fold, and lower than model and satellite-based exports are about 3 to 7-fold. So neither models nor satellites are reproducing export trends in the Pacific. And the reason I want to bring that up is because we are, we're so certain that burning of fossil fuels and accumulation of greenhouse gases in the atmosphere and the ocean is going to impact the habitability of our planet, that we are more and more willing to perturb this ecosystem to buy us time. So I was a co-author on the National Academy Consensus Report of Marine Carbon Dioxide Removal, which spanned things like seaweed aquaculture to artificial upwelling to alkalinity enhancement to iron fertilization, which is an old story, but a new story again, as well as ecosystem restoration. And this work is moving forward. In the US, the ARPA-E program announced $45 million for development and frameworks for monitoring, report, and verification of marine CDR. And not too very long ago, the Meta announced $50 million for ocean alkalinity enhancement. And there's been sessions at major international conferences as well, workshops for exploring iron solution. And when I was at this workshop, it was really striking to me that there were a lot of senior researchers, people who spent their whole lives studying the ocean, helping to build many of the baselines that I've already shared with you. And despite this commitment, really, to the oceans, they are still willing to further perturb these ecosystems. And that's striking because ecologists don't usually sign up to tinker with nature. And we are in an inflection point here in the US, where I think we need to move from inaction to action, get people more invested. I know this is not the same across the globe. Here in the US, we split across political parties with only about half of Americans even viewing climate change as a major threat. And that despite the fact that this is, as I said before, real and measurable change. This is not political at all. These are high quality QAQC data on how our oceans are changing. So I will end with this last point. I think I'm coming in on time here, but time series are sentinels of ocean change. They are showing us the ways in which the oceans are changing, and those are clear and measurable. And there's a part of the story that's hard to tell. It's a little bit heartbreaking and hard to watch. The time for action, I do believe, is now. And I think for us all to be on the same page, those of us particularly in time series need to start telling these climate stories more differently and more widely. We need to bring our whole self because we do care about these ecosystems quite deeply. And with that, I will end, I think right on time, and say thank you for believing in the critical role of time series. This is our last get together for most of the folks that are working with HOT, where we went to escape room and we did escape.